Great, everybody. Well, I'm Betsy Rothermel, the program director for herpetology and restoration ecology here. And we have our speaker today is Andrew Mularo, who you've probably seen around for the last couple of months. We finally reeled him in to give a seminar. So Andrew completed his bachelor's at Ohio State University in evolution and ecology in 2019, and he entered a PhD program at Purdue University. And I noticed his background includes a lot of excellent teaching and mentoring, and some of that he's going to be showing you the results of for the research mentoring he's done. He's also been doing a lot of great outreach programs over the years, going back college, high school. Is everything okay? Okay. And I had it. These were intriguing topics, especially to me as an amphibian ecologist. So some of the titles of his programs were A Totally Unforgettable Time on Land Without Lungs. Okay. Anyway, so that was cool. And we were really fortunate in our lab to scoop up Andrew as a postdoc for this NSF-funded study of mechanisms of genetic rescue, which is based on experiments with these experimental populations of inbred and gene flow mosquito fish. And he's been here since January, plugging away on the last, well, keeping the mosquito fish alive, plugging away on the last set of experiments on critical thermal maxima. And he's going to be here another 2 or 3 weeks before he heads back or up to Michigan State University, where he'll be jumping into the lab part genomics work for this project. And today, though, he's not talking to fish. He will be presenting results of an aspect of his dissertation research. It's very relevant to where we are here in Florida because it involves frogs and toads that are introduced to Florida. So I guess I'll just turn it over to Andrew. And you can say your title and go from there.
Can everybody hear me okay? And if anyone can't hear me on Zoom, Laura, would you just let me know to speak up? So yes, I'm Andrew Mularo. I am a relatively new postdoctoral researcher at Michigan State, and I am going to talk to you today about the environmental drivers of invasion success in Florida's introduced anurans. And so, as Betsy said in her introduction, I'm actually down here studying these wonderful little Gambusia holbrookii mosquito fish. And so if any of you have kind of wandered behind the maintenance shed and have seen all of these ponds, they're filled with hundreds of mosquito fish that have been subjected to different evolutionary history treatments. If you see me there, stop by and say hi. I've pretty much been talking to fish the entire time I've been down here. Not a lot of people talk. But I unfortunately just got started with this, so that's not what I'm here to present on today, but I figured some people might be a little bit confused of how I went from amphibians to fish and all this kind of stuff. And really, in principle, I've worked with a bunch of different species across broad taxonomic groups, tiny frog-biting midges in the neotropics, leafcutter ants, Massasauga rattlesnakes, lesser prairie chickens. And so I've been able to kind of get a diverse experience working with a bunch of different species. But my entire research theme is centered around this concept called the extinction vortex. And this might be familiar to some folks, but for those that are not familiar with it, the extinction vortex is a phenomenon that is frequently observed in nature where different pressures are put on a population to reduce its population size. And once a population becomes reduced, the frequency of inbreeding increases, there's a reduction in adaptive potential, and effectively it leads to this negative feedback loop where it's really hard to kind of escape all of these deleterious effects, and ultimately the population goes extinct because of that negative feedback loop. And so all of the species that I've worked on have had this similar theme of going through a reduction in population size and experiencing the extinction vortex. But this topic, I don't know, when I was in grad school, it got really depressing. Like, it happens a lot, but it's so depressing. It's important to know about. But I became really interested in trying to figure out ways in which a species or a population can actually escape the extinction vortex. And it turns out that there's a lot of different ways. Gene flow and genetic rescue, which is what I'm currently studying with mosquito fish, rapid adaptation to climate change, different biotic interactions, selection on viability. And so these are the different themes that I actually evaluate in my research. And that brings me to what I'm going to talk to you about today, which are invasive species. And I consider invasive species to be the quintessential example of escape from the extinction vortex. Because if you think about it, an invasive species is transplanted from its native range to a new region. So there's that reduction in population size. Yet all of the ones that we observe as successful invaders have been able to overcome those challenges in one way or another and successfully spread. And so I became really interested in studying the mechanisms of invasion success as they relate to this kind of reduction in population size. To kind of frame this in a way related to the extinction vortex and kind of recapitulate what I just said. So the genetic paradox of invasive species, they undergo a population bottleneck or reduction in population size, which ultimately leads to a reduction in genetic diversity. There's another hypothesis built into this concept called the central marginal hypothesis that says once an invasive species is introduced, it undergoes iterative population bottlenecks as it continues to spread throughout the range. And so that is expected to further reduce genetic diversity. But despite all of these challenges to their evolutionary biology, we see population persistence and expansion with most of these invasive species that we study. And this can be for a variety of different reasons. And so one of the main ones that is evaluated are multiple introductions and propagule size. If you have many individuals coming at the same time, that can bolster genetic diversity and establishment success. And so this is an example of one species that everyone's probably seen, the brown anole. So it was introduced into Florida in the late 1800s. And genetic research done by Kolb et al., 2004, so quite a while ago, showed that there were different genetic lineages that were introduced from their native range, which is what these different colored circles are, so the different colored haplotypes. And as you can see, all of those haplotypes are also present in different ratios in Florida. And so what these authors suggested is that multiple genetic lineages were introduced into Florida at multiple different times, which is likely to have bolstered their success as an invasive species. And in addition to this propagule pressure, or this propagule size and genetic diversity, variable environmental conditions can also increase establishment success. So this is a species that's near and dear to my heart because it's coming to Michigan, actually, which is the spotted lanternfly, which was introduced on the East Coast in the harbors. And what has been shown across many different aspects of studies in this species is that they have a broad tolerance to a variety of different environmental conditions and are expected to spread because of this tolerance to a broad array of environmental pressures. Just kind of throwing out this picture that my dad sent me because he keeps squishing them at work. And he's like, I squished another one today. So that's great. So we have propagule pressure and increase in genetic diversity, but also a broad tolerance to a variety of environmental conditions that are ultimately the drivers of invasion success. But a lot of times, these factors are really unknown, and oftentimes it's too late to study them once an invasive species becomes established. It's already causing problems, and it effectively is too late to implement any new management techniques. And so to understand the success of invasive species, one must consider the history of the invasion and the environmental conditions in which they are established in order to make better predictions for any future introduction and establishment events. And I'm sure I don't need to tell people that have lived here in Florida a while that Florida has a ton of invasive herpetofauna or reptiles and amphibians. So in 2011, Walter Mashaka published a gigantic monograph detailing accounts of the invasive reptiles and amphibians, found 47 species that actually increased exponentially since the first invasive species, brown anole, in the 1870s. I recently looked at iNaturalist and found that there were actually 71 non-native reptiles and amphibians that had more than one observation attributed to them. So more likely than not, there's either multiple people releasing them or they might be established. And so this is likely an outdated number, and there's probably a lot more invasive herps that we don't actually know about. And why does all of this matter? Well, you know, they're really cute. I like the brown anoles. I like the Cuban tree frogs. I like everything. It's caused some extensive damage to the native fauna of Florida. And so this is not meant for everyone to read in detail, but this was a literature account that took dissections of Burmese pythons and counted each individual species of bird that was present in the gut and bolus of the Burmese pythons. So yeah, we've got American coots, purple gallinules, domestic chickens even, little blue herons, snowy egrets. They're eating a ton of different species of birds. And they've even had some effects on our charismatic megafauna. So this was from the Everglades, where a Burmese python attempted to eat a full-grown American alligator and literally exploded, resulting in both of their demises. And so that's just from the predation side is a really bad thing that these herpetofauna do. There's also the potential for the spread of diseases. And so this is a microscope of dissected Rayletelia orientalis, which is a parasite that's present in these brown anoles, but can actually be transmitted to native snakes and lizards as well. And so these invasive species not only are eating and out competing the native counterparts, but they are also spreading diseases. And a lot is known about these ecological repercussions for some of these species. But unfortunately, when they're here, we really don't know exactly where they came from. And the majority of accounts of like the history of introduction and where they've been spread to are reported in very scant anecdotal literature accounts, which makes predictions of where the most likely areas of spread are to be incredibly difficult. And not saying that these aren't important accounts, but there really hasn't been any systematic way to address how these species are being spread and what their ramifications are, at least in the early to mid 1900s. And so that's kind of what I became interested in as it relates to the Florida herpetofauna. And I became interested in these three different species of frogs that have been introduced into Florida at various time points. So we have the cane toad over here, the greenhouse frog, and the Cuban tree frog. And I'm sure Betsy and I caught a gigantic one a couple weeks ago near my fish ponds. We thought it was eating the fish. But these are pretty widespread throughout Florida. And this represents a good opportunity to do some comparative studies to see what are the drivers of the invasion success, what is the history of invasion, because the invasion histories of these species are poorly characterized. There are many gaps in knowledge of the environmental drivers of the invasion success in these species. And probably most important of all, the ecological ramifications to Florida's native fauna are mostly unknown in these species of frogs. There's scant reports of out competition and predation, but mostly unknown. And so that's what leads me into my research aims of these particular studies that I conducted for my dissertation, as well as some undergraduate advisees. The first aim was to reconstruct a comprehensive invasion history of these invasive anurans of Florida. The second aim was to describe the environmental conditions that facilitated the successful establishment of these invasive anurans. And then my third one, which is really hot off the RStudio right now, like very new data, is to evaluate how habitat features influence interactions between invasive anurans and their native counterparts. And so the first aim, to reconstruct the comprehensive invasion history, we have been very lucky in the last couple of decades to have an explosion of public data repositories that actually keep good records of the occurrences plus when and where they were found of all of these invasive species. And so what I actually did was I combined two different data sets on these three species from public repositories, including the United States Geological Survey, sorry, and then VertNet, which is a subsidiary of the Global Biodiversity Information Facility, in order to get a comprehensive collection of dated occurrence points of all three of our species in an attempt to figure out where they were introduced from, where they spread to throughout the history of their invasion. And for each species, we got several thousand data points. This is such a great resource, but it's really hard to kind of go through each individual data point and figure out where it was and create the timeline that way. And so we actually designed a spatial model to be able to generate predictions of where the most likely spatial occurrence within Florida was at each different decade since their introduction into Florida. And so this technique is called empirical Bayesian kriging. And basically what it does is when you have a data point, it takes in the values of the top 15 data points that are around it and generates spatial models for the best fitting value in those empty spaces around it. So you can effectively predict what the likely value is for an empty space that you define. And it also gives us a good estimate of error. So what areas are we most confident in? This is what this plot is down here. What areas are we most confident in, and what areas are we least confident in with where they were introduced to? And so we extracted the area interpolated based on this kind of heat map of time effectively by decade to actually measure the trajectory of where they were spreading into since their introduction. I'm going to kind of go quickly here, and it's a little hard to see the time with this text down here. But the cane toad, it was actually a really interesting result. So it was introduced in the mid-1950s and had a previously undocumented introduction on the West Coast near Tampa and have since spread outward and have gotten kind of up north, spread throughout the state, and then in the most recent decade since 2022, it appears to have undergone a contraction. But what that is facilitated by is actually several of the toads were introduced into the northern panhandle and were frozen out effectively. So they ended up going extinct and were unable to establish in that more northern region. And so this is the current predicted distribution of cane toads based on our study. And it largely corroborates what's known from Mashaka's work and the Florida Fish and Wildlife Service. So we got West Palm, Miami, not in the Everglades, cane toads do sink. That's the price of being such a big, large toad. Now let's look at the Cuban tree frog. So the Cuban tree frog was introduced at around the same time, largely focused in the area of Miami and then in the Florida Keys, which matches what these early literature accounts, but actually established nearby where we are right now, kind of around Okeechobee, Venus, but then quickly by 2000 was present in the Panhandle and now occupies the large majority of the state of Florida. So we see Cuban tree frogs everywhere. They're fairly cold tolerant and are continuing to spread into the northern regions. For the greenhouse frog, Eleutherodactylus planarostris, this was actually first introduced in the early 1900s, again around Miami, and you can't see the keys there, and has had a really patchy distribution and series of spread. Now, this species is very small and hides typically in flower pots and potted plants. And so it is largely facilitated through the trade of these, the expansion is largely facilitated through the trade of these plants. And this is the current estimate of their distribution, which herpetologists have looked at this and said, that's probably not true. And the reason why is that these guys are incredibly hard to find. They are undersampled. They are less than 2 inches. They hide under pieces of wood. They have a very small call. And so more research is needed to prioritize where these guys actually are in modern day. And the records are largely centered around urban areas where people are most likely to encounter. And so to kind of summarize what's going on here, what we see are these range expansions in the cane toad and Cuban tree frog. But the cane toad underwent our contraction, meaning that they spread into the northern region and then just failed in the subsequent decade. Cuban tree frog continues to expand at an exponential rate, and current records suggest that they are in Georgia, in Louisiana, in Alabama, and South Carolina now. And so that's our troublemaker right here. Now, the greenhouse frog is largely undersampled. It has the model predicted that it underwent a slightly less exponential growth, kind of a leveling off. Again, this undersampling of the species is really a problem with the eleutherodactylids because they are so small. And so kind of major takeaways of this, which ended up being my second chapter of my dissertation, is all invasives likely had multiple jump dispersal events. So they were introduced into Florida multiple times and had different kind of like hodgepodgey interactions or introductions throughout the states. There were previously undocumented introductions of cane toads on the West Coast, which suggests multiple introductions from their native range in South America. Cuban tree frogs, like I said, are the troublemakers. They appear to be the only one with continuing expansions in the state. The other two species appear to be occupying where they're going to occupy for the foreseeable future, climate change. And then the caveat that I've explained here is that greenhouse frog is likely an undersampled species, and so more work needs to be done on figuring out the distribution of this particular species. So that is my attempt at a comprehensive invasion history of all 3 species. But now the second aim was to describe the environmental conditions that facilitated the successful establishment of these invasive anurans. And this research was largely, or actually I should explain the background first. And so there's been some research in this topic already. So actually my current colleague, Cinnamon Mitten, at Michigan State right now, did some work on cane toads and showed evidence of acclimation towards colder climates. And so even though the individuals that were present in the northern region ended up going extinct, we expect that might change, possibly. And then Cuban tree frogs are predicted to expand their range throughout the United States as climate changes. So Dennis Roeder has run multiple species distribution models for Cuban tree frogs under future climate change projections. And if it continues by 2080, they're expected to be in Texas, Louisiana, parts of Mexico, potentially back in Cuba too. And so, yeah, this species is going to be a real problem. But there's still gaps in the literature, and not a lot is known about many other different environmental drivers of the invasion success. And so I was very fortunate to work with 2 undergraduates that really became interested in, okay, they're established here. Why have they been so successful? And so 2 of my undergraduates for their honors theses picked different research topics for their honors theses. And so I'll kind of summarize their results in the next couple of slides. And so the first one I wanted to talk about was salinity tolerance in the Florida tree frog. So telling the background story of this, so this is an all familiar mangrove forest. And Addie and I actually came down to Florida in 2022, and we were putzing around these coastal islands that had mangroves on it, headlamping, not really paying much attention to, anything in particular. We were just out looking. And what we ended up finding was large, massive Cuban tree frogs that were perched right above the salt and brackish water that were present in these mangrove forests. And for a frog biologist, this was really weird because frogs have very porous skin. They absorb everything, which makes them very sensitive to pollutants and also very sensitive to salt because salt is dehydrating and water is expected to be expelled from them at a greater rate than other animals with thicker, less porous skin. And it was really interesting that, and this is just anecdotal, that they were huge. Like they were just so massive. And so then Addie and I kind of were thinking, like, do these frogs have the ability to actually develop tolerance for salt water? And that was an important question because, as you saw from my invasion history, they were introduced into the Keys and along coastlines, and so they're very likely to be able to, they're very likely to be exposed to this salt water. And so what Addie did is actually our collaborators were very generous and gave us 24 Cuban tree frogs collected from Gainesville. We took them back up to Purdue. And we wanted to test if we can actually induce a physiological response towards the saltwater stressor in Cuban tree frogs. And so we divided half of the individuals into a control treatment where we kept individuals in an aquarium that had paper towels and fresh water, and the paper towels were dampened with water that was 0 ppt salt solution. And this was changed every day, every other day, depending on how much of a mess these Cuban tree frogs made. But then the other half, we set them up in the exact same way, the exact same type of aquarium, but we dampened their paper towels with 8 ppt salt solution, which is reflective of brackish conditions typically found in mangrove forests. And so every day, this experimental treatment was just absorbing this brackish water solution. And we wanted to test if the experimental group had a higher tolerance of salt near the end of this experiment. And so in order to do that, this is a little difficult to see, at 4 different time points of this experiment, we exposed all individuals to an acute salt exposure, meaning that we submerged them in 8 ppt solution salt for about 6 hours and measured the amount of weight that changed pre and post acute exposure, with the expectation that the experimental group would be able to develop a greater tolerance to the acute exposures than the control and lose less weight because less water is being expelled. And so these are our results, which we recently published. I'm very proud of Addie in the Journal of Experimental Zoology. And so this is the control group. So the x-axis are the 4 different time points. The y-axis is the percent weight change, with a positive value indicating a gaining of weight and a negative value indicating a loss of weight, which is what we expected for a lot of these cases. And so, as you can see, the vast majority of individuals lost weight when they were initially exposed to salt and continued to lose weight each time they were exposed to this acute salt exposure. A couple gained weight. We're not sure why. I don't know. It's still being figured out. The reviewers didn't ask too many questions about that. And then the experimental group was actually really interesting. So at week 0, the first exposure, they lost about the same amount of weight as the control group did. But then at week 2, there was a dramatic amount of weight loss that we observed in this experimental group. So they lost double to triple the amount of weight that the control group lost. But at week 4, about a month of being exposed to the salt treatment, there was a statistically significant rebound, which means that these individuals, even though they lost a tremendous amount of weight in the week prior, or in the 2 weeks prior, they actually lost less weight in week 4. And so what this was suggestive for us is that long-term exposure after about a month can potentially induce a recovery of weight loss after being exposed to salt. But unfortunately, it didn't really last very long. So after about 6 weeks, it became statistically insignificant, unpredictable how they were going to lose weight. And so this was really interesting. And what this actually showed us is that acute minimal exposure to brackish water can potentially induce a physiological plastic response in their ability to tolerate salt. So that was Addie's project, which we're very excited got published. But now I wanted to move on to Jack's project, which is currently in review in biological diversity. And he became really interested in our beautiful little Eleutherodactylus plant rostris. Again, really tiny frog, very understudied. And he was interested in the climatic shifts that were potentially displayed by this particular species. But now, for any of the herpetologists in the room, Eleutherodactylus is one of the most species-rich genera of frogs, over 100 species. And we actually have an opportunity to compare climatic shifts not only in our single species, but in 2 other species, the Lesser Antillean whistling frog and the Puerto Rican coqui frog, that have also become successful invaders outside of their native range. And the reason why this is important is that it gives us a broader evolutionary look at how these species respond to different climate conditions. And so for this particular paper, what Jack and I did is we actually did a similar thing. We did a systematic reconstruction on a global level of all of our species at where each one has successfully established, has recently been introduced, but we don't know if it became established or not, as well as a failure to establish, meaning that they're not found there. And so the greenhouse frog, it's all over the United States, but it's actually been introduced into Japan, the Japan area, China, Malaysia, failed in South America. So there's a lot of variability. And this is the most broadly distributed frog. So interesting different climate conditions potentially that this species is exposed to. The Lesser Antillean whistling frog has also been very successful. So it's native to the upper Lesser Antilles, but it's introduced into South America, all these different places. Has recently been introduced into the Czech Republic and Germany. We'll see. I don't think it'll be old there much longer. And then the coqui seems to be the one that's most restricted, having only been established in the Virgin Islands as well as Hawaii. It has been introduced here in Florida, but it's not established here. It is very, anecdotal reports say it's very sensitive to cold and freezes. And so it hasn't yet become permanently established here in Florida. And so we have these 3 species that are very closely related, that have very different invasion histories. How do each of these respond to the different climate conditions? And are there any potentially evolutionary conserved traits that make these ideal invasive species? And so what Jack did is he actually downloaded several thousand data points for each species from the Global Biodiversity Information Facility, and was able to pair all of these occurrence data points with corresponding temperature and precipitation conditions present in the World Climate database, which is a really comprehensive database of historic, current, and future climate data that I've been really loving working with the last couple of years. And so with these results, we actually were able to generate a ton of different variables and plot them on a principal component analysis for each species the principal component analysis, for those that may be new to these types of statistics, is basically a technique which takes a bunch of different variables and distills them down into arbitrary variance axes to basically get at, are they separated, clustered together amongst all these different variables. And in this case, we had 19 different climate variables that were assessed. And when we put each of these species through a principal component analysis, what we actually observed is that the non-native in blue regions were significantly more variable in their established regions than what was present in their native regions. They seem to have a very close, clustered, restricted niche in their native range, but are able to expand out into a bunch of different climate conditions. And that's true of all three of these species. So that's really, really interesting that it's kind of all the same. Specifically, we looked at annual mean temperature and annual precipitation as just a couple of the different variables to highlight. And we observed significant differences in annual temperature between their native and invasive ranges. And in particular, they were able to successfully establish in much colder regions than what was present in their native range. Their native ranges seem to be very, again, narrow in the amount of temperature variability that they have. Precipitation was a little bit more difficult to predict, but overall what we're seeing here is, especially for Planerostris and Johnstonii, the whistling, Lesser Antillean whistling frog, is that they tend to establish in regions that have higher annual rainfall than what is present in their native range, which shows their reliance potentially on humid environments for their establishment success. With Koki, it was a little bit smaller, so maybe they're able to tolerate it a little drier, and it was significantly different, but it's not anything to really write home about. And so these are kind of the main findings from all of our undergrads research projects. And so just kind of takeaways here. Physiological adjustments to saline conditions allow Cuban tree frogs to tolerate saltwater ecosystems, is what our claim is for the physiology experiment that we conducted. And then based on the results of our climate analysis, what we're suggesting is that in the genus Eleutherodactylus, it is evolutionary conserved to be able to tolerate colder temperatures than what is present in their native range, as well as variable precipitation, potentially a reliance on more humid conditions for at least 2 of the 3 species. And so this was kind of our contribution to some of the gaps in the environmental drivers of invasion success in our species. Then we had this third aim, because none of this, I mean, it's cool from an evolutionary biology perspective, but what people are really interested in is the interaction with native species. And since I've been here at Archbold, I've been seeing a lot of real cute native species: squirrel tree frog, the pine woods tree frogs, which I think are my favorite, and southern toads up at Red Hill. I don't know if any of you have seen them. And so Florida has just a bunch of different native species that are potentially threatened by the presence of these 2 invasives. And we all have seen this kind of image of these native frogs gathering towards light to be able to collect and eat all of their insects and stuff like that. Leaves them pretty vulnerable to predation by our bad guys here. This was a photo taken by Steve Johnson and colleagues at the University of Florida of a Cuban tree frog eating a squirrel tree frog. It's a little unclear how can they do this, but they can. And Cuban tree frogs can get ginormous. Like I said, Betsy and I caught one. It was eating a gecko, so not our fish, but that's still kind of interesting. And so there's a big threat that these invasive species pose towards our native anurans. And these species are really abundant in urban environments where we're seeing these native species gather under light poles and stuff. Here's one of the gargantuan Cuban tree frogs, cane toad, that was resting in a cinder block. And so these invasive species are absolutely encroaching on the habitats that we typically find native species. And so that's a potential problem. But really, there's not a ton of studies to evaluate what the actual drivers are of these interspecies interactions and if they actually do pose a significant threat to the native anurans. And so I had two undergraduates that were really interested in this kind of phenomenon because it appeared as though that these native species of frogs tended to use urban environments to help them collect and eat insects, but also that cane toads and Cuban tree frogs were really abundant in urban environments. And so these two undergrads of mine were interested in evaluating urbanization as a potential driver of interactions between native and invasive species. So I mentioned at the beginning of my talk, this data is like days old. And so we're working on getting this manuscript ready. And I'm really looking forward to talking with some of you about maybe how to refine and improve these analyses or any other suggestions that everyone might have. So to compare or to evaluate whether urbanization is a potential driver of interactions between these, we again went to GBIF and obtained modern occurrence data for all of the native species of frogs, I should say tree frogs and toads, and our invasive counterparts, cane toad and Cuban tree frog. And we were able to use ArcGIS to actually match these occurrence data points with the USGS National Land Cover Database, which measures the percentage of urbanization or percent impervious surfaces that are present throughout the United States. And so we were able to get basically a mean percent impervious surface for each individual occurrence data point. So like, were they in a moderately urban environment, a completely urban environment, or a totally rural environment? And what we also did was we had that data and we needed to basically figure out how close the species were to each other and whether the urban development was facilitating that. And so in order to do that, we ended up calculating the geodesic distance between the native species here in blue and the closest invasive species documented within that year. And basically for each native species, we ended up with a percent impervious surface that they occurred at, or percent urbanization, and the relative distance to the closest documented invasive species. And so I kind of wanted to show these preliminary results to everybody here. And so on the x-axis here are just the overall mean impervious surfaces, so the degree of urbanization. 0 means completely rural, no urban development. 80% on the high end is near cities, really urban, not a ton of wild areas. And so our two invasive species, Cuban tree frogs and cane toads, occur at the highest level of urban development. And that's not entirely surprising. That's pretty consistent with what people have observed. And on the lower end, I have to read that over here, this is Dryophytes avivoca and Hyla femoralis. So the pine woods tree frogs that we're seeing here don't seem to tolerate urban development well at all. And avivoca is not common in Florida to begin with. So anyways, this was a particularly interesting finding, kind of confirming what we expect with invasive species. Now let's take a look at the distances and kind of covering everything up. So I'm just going to need to describe it. So on the x-axis here are the square root transformed mean impervious surfaces. So again, continuing to the right would be a higher degree of urbanization. And then the relative distance to the closest invasive species. And so for the green tree frog, Dryophytes cinereus, we see a moderate negative relationship between those two variables, which indicate that as urbanization increases, the native frogs are being driven in closer proximity to the Cuban tree frogs. It's a pretty weak relationship. I mean, Cuban tree frogs kind of are everywhere, and so it was not particularly strong. We talk about squirrel tree frogs, which we see a lot around here. There was another negative relationship with this, but it's very zero inflated, which basically means that across a great deal of habitats, they're just in very close proximity to one another. But it does show a moderate trend to suggest that urban development is again pushing these species in closer proximity to one another. When we look at southern toads compared to cane toads, we also see the same thing. You know, cane toads are also very good at living in urban environments. Southern toads a little less so, and so as urban development increases, we observed a moderate decrease in the distance, again, being brought into closer proximity. And so this is really interesting. We're still kind of wrestling with these results and like how powerful is this trend actually. And I'm only showing these three species because the other species that we had in our diagram had a very, very low sample size that we were unable to run any relevant statistical model that would be powerful enough for any inferences. But Ireland, Desi, and I kind of asked, is this actually the main driver of what's bringing these species in closer proximity or spreading them farther apart from one another? And urban development is not the only relevant factor here. And so these are the new data that we have where we put the urban development as well as all of our climate variables that we collected from the World Climate Database into a PCA. And again, distilling that down to some arbitrary variance axes. And what we can see, so the pink here is the Cuban tree frog, Osteopilus septentrionalis. And then on the far left-hand side here, we have the green tree frog and some of our squirrel tree frogs that seem to occupy a slightly different ecological niche than what the Cuban tree frog is occupying, at least on principal component 1, which is about 30% of our variance. So that's really interesting. We still see quite a bit of overlap, but there's some differentiation. When we look at the toads, we kind of see the same thing too, right? So it's about the same amount of variance. Cane toads are clustered over here. And then especially our southern toads, there's overlap, but they are spread apart from them. So occupying a potentially slightly different niche space than the cane toads are. And so that's really interesting. That led us to ask this question, okay, what's driving these? And so we actually plotted the loading scores of each variable that was incorporated into our models to see what is separating these different clusters. And what we actually found was that this thing called bio14 here is present in separating the clusters between native and invasive species for both tree frogs and toads. And what is that? The precipitation of the driest quarter of the year. So in the driest quarter, how much precipitation there is effectively seems to be the main driver of this niche differentiation between native and invasive. And so this was our kind of new focal variable to investigate because mean impervious surface didn't really show much differentiation at all. And so when we look at this, precipitation of the driest quarter was actually the one where we observed the two invasive species occupying the most dry niche spaces. And then all of the other invasive, all of the other native species seem to occur at wetter regions of Florida and the American Southeast, with our two highest precipitation individuals being Chrysoscleus, which is the Cope's gray tree frog, as well as our cute but sadly very absent on Archbold eastern spadefoot toads, which seem to rely in these dry seasons on a higher level of precipitation than what the invasive species can tolerate. And we plotted the same thing, the distance, but against the precipitation of the driest quarter for our tree frogs to see if that is what is actually separating the two species. And unfortunately, again, pretty new data, but for the tree frogs, there doesn't seem to be a very big relationship. It seems like distance to the nearest invasive, it doesn't matter what the precipitation in the dry season is. But the most interesting one was the southern toad. So it does suggest here that these southern toads, which have a positive relationship between the precipitation of the driest quarter and the distance to the nearest invasive species, that occupying wetter regions in the dry season is a potential safe haven for these southern toads to not have to interact with the cane toads. If it's a lot drier, they're much more likely to encounter and potentially be predated on or outcompeted by the invasive cane toads. So like I said, all very new data, and I'm looking forward to maybe chatting about this more in the question session. But some of the major takeaways, here's one of the cute oak toads I found a couple days ago. So increased urbanization may put native species at greater risk of encountering their invasive counterparts, but the relationship that we can tell from these data sources is relative. However, precipitation within the driest quarter of the year shows the strongest differentiation in the climatic niches between native and invasive. And so that is likely the key variable to watch out for with these interspecies interactions, is how much precipitation is in a region for the driest of the year. And so to kind of end my talk here, I can take some questions. I hope I've convinced everybody that a wide array of invasion histories, tolerance to a wide array of environmental conditions have led to the successful establishment of the 3 invasive anurans of Florida, as well as other species that we kind of pulled into our study. And I would argue that comparative studies of multiple species within a genus and across a landscape is key to disentangle the broad drivers of invasion success and any potential ecological ramifications that are posed by these invasive species. And I will acknowledge people that helped me. We came down in 2022 to see if we can observe these invasive species. We found lots, and so that was a real nice treat in the middle of COVID to be able to be down here looking at cane toads. And so lots of funding to support this, lots of co-authors and people that helped with this. I'm happy to take questions.
Thanks, Andrew. That was a really, really cool talk. So I wanted to ask a question about the first half of your talk. Do you why did the cane toad and the Cuban tree frog wait this long to invade North America? If they can survive everywhere, what was keeping them from coming here?
You mean in when they were introduced initially in the 1950s?
Yeah, I mean, they've been around for millions of years, and there have been times when they could have easily come over.
Yeah, the answer is humans. And so Cuban tree frogs were thought to be introduced through the nursery plant trade, which a lot of these nurseries originated in Cuba. So they were thought to be stowaways as that market continued to increase. As far as the cane toads, the original documentation, 1955 in Miami, were pets. So people that had them as pets, and they arrived in the Miami airport, they ended up, at least the record says that they escaped into the canals and spread forward from there. There was a failed attempt at introducing cane toads from Puerto Rico in the 1930s as a potential biocontrol of sugarcane pests. That was not successful on multiple fronts. They didn't eat any of the pets, and then a cold snap hit, and they were all extinct. And so these species have relied on humans for their transport. Yeah.
So, I mean, these Cuban tree frogs, as you showed, are super salt tolerant. And there have been times in the glacial or whatever ecological past when there have been. So do you think they've colonized and then gone extinct over the past whatever several 100,000 years, and then are now recolonizing as climates change?
It's certainly possible. And the Cuban tree frogs, particularly being good as stowaways, can drift into new regions. But again, I think it's about that propagule pressure too. So with human trade, they're being brought in more and more frequently, which increases the percentage of successful establishments. Yeah.
Yeah, thanks for the talk. I was really intrigued by the introduction where you spoke about kind of how you got to this topic is extinction vortex and the idea of introduced species as the inverse of that. But I'm not quite sure if I understand how you tied it together at the end. So could you maybe restate or expand a little bit? What have you learned about the few of many introductions that survive that tell us something about why things go extinct?
Why things might escape the extinction vortex? Yeah, so I think that invasive species, at least what I've learned, because you can't tell when a species comes to a new region if it's going to be a successful invader or not. You're already studying it when it's too late. And so what I've learned, I guess, is that this broad tolerance to environmental conditions, the ability to physiologically adjust and potentially adapt to new climate conditions, as well as, again, this propagule pressure of having multiple different introductions and multiple events of spreading throughout their new region is what's promoting an increase in population size and diversity to be able to escape being extinct.
So it doesn't necessarily have a lesson for conservation management, say, because it's really about the intrinsic properties of the species and how fungible their environment can be for them to succeed in. Is that right?
That's what I would. Yeah.
Great talk. Kind of on that note, I'm curious about how you square away the observation that invasive species have a broad sort of environmental niche in their invaded range versus a narrower niche in their non-native range versus their native range, excuse me. Do you think that that is related to traits or population history? I'm not really sure how that makes sense, how they can be so adaptable. Then why are they not taking over within their native range?
Yeah, so this has been a pretty hotly debated topic about what the actual niche of an invasive species is. Do we consider what's present only in their native range as their real niche that they've then expanded, or do we take everything into account when it comes to their whole distribution, native and non-native, as part of their niche? These species exist on islands, a very small region that only experiences a little bit of fluctuation in habitat characteristics. And so it's hard to tease apart, are they intrinsically tolerant of that? And is it part of their niche and they just haven't had the opportunity to expand into it yet? Or does that represent evolutionary responses to a new trait that they previously weren't tolerant of? And to get around that is experiments like common gardens, exposing them to different regions. There's been some work on that, or I'm sorry, different climate conditions in a controlled setting. And there's been some work, but not.
Gotcha. So if I'm understanding correctly, it seems like dispersal limitation is the main thing, that they're not really able to experience the true range of conditions.
Yeah.
Thank you.
Great talk. I thought it was really interesting to see how the Cuban tree frogs are really expanding over time, especially into freezing areas. I was wondering kind of what's special about the Cuban tree frogs? Why are they so good at invading? And how are they surviving freezing temperatures and salt, everything?
Yeah, so I can't really speak to physiology behind freeze tolerance in them, to my knowledge, the literature is just like they can tolerate, but the mechanism is still a little bit unclear. Behaviorally, though, they do tend to tuck away and find shelters to avoid exposure to some of the most grueling conditions. And so in a lot of cases, they're in PVC pipes and in and around people's homes, in tree bark and holes. And so they're able, it is my guess that they're able to behaviorally avoid a lot of these deleterious conditions, whereas cane toads being a lot larger, for example, it's a harder time for them to do that. But at least the physiological, yeah, I don't know. Yeah.
Thanks.
Yeah, awesome talk. So sometimes the line between an introduced species and a truly invasive one can be a bit hazy, but I noticed that you've included greenhouse frogs in your talk. Do you think that it's the fact that they haven't been studied enough, or maybe their impact is on a very small understudied species? Do you think that there's something that would make them invasive and worthwhile studying, or do you think it's they probably don't have much of an impact because it hasn't been seen yet?
Yeah, this came up in peer reviews, so I know how to. Yeah, so there's two different, at least broad definitions of how we classify invasive species, and most people tend to. And I'm going to go all the way back to my first chapter, or for my first part here. Most people in the literature will classify an invasive species based on impacts, like ecological, economic. Do they cause damage? But there's another subset of the literature that I subscribe to that suggests once they hit this exponential growth phase of their spread, that is what the line is between an introduced, where they're just kind of muddying around and not dispersing much, versus once they've hit that exponential growth rate. The reason why I subscribe to that is when working with these types of data, it's hard to quantify impact, but you can quantify spread and population growth a lot easier with those types of data. And so for the purposes of this paper, we considered all three as invasive because all three of them have hit that aerial growth or exponential growth in an occupied area, which showed that they're spreading very widely throughout the state. Did that answer your question?
I believe so. Can I expand?
Yeah.
Is it your assumption then with that definition that once there is that exponential spread, there is some sort of negative impact in some way, or there will be, even if it's not seen yet?
Potentially. Yeah, potentially. Or does the definition of being an invasive species have to have negative interactions to begin with, right? That's kind of what some of the authors have challenged in that literature, is like, it doesn't have to have a negative impact to be invasive as long as it's just widespread. So yeah, it's complicated.
For sure, yeah.
Yeah, I have one online. Yeah, okay. So this will be our last question. It's online from Tiffany. Let me get back to my computer. Okay, Tiffany says, I know you didn't study urbanization effects for greenhouse frogs, but their invasion doesn't seem to me to be related to urbanization. I catch them daily at APAFR, but only in good flat woods, scrub, and prairie far from buildings. How do you think they were able to integrate so seamlessly into natural habitats?
If I understand the question, and let me know if I don't address it fully, I think there's a lot of different factors. They are a direct developing frog. They don't rely on water, and they lay their eggs in a variety of different soil types. And so this avoidance or this lack of a need to go back to water sources has probably led to their vast expansion into regions unoccupied by humans. Small size. Betsy and I have discussed this. They use gopher tortoise burrows and a lot of other natural species burrows to shelter in. And so I think they're just very good at detection and very good at dealing with deleterious effects of habitat and other climate aspects. Yeah. Did that answer your question, Tiffany?
I'll let you know in a second.
Sure, yeah. Any more?
Yes, thank you.
All right, perfect.
Good. Thank you for joining us online, everybody.